Hi everyone. Welcome to the Science Info channel. Guys, you must have heard about the brain. Have you ever heard about mini-brain? If not, then today we are going to discuss about the mini-brain. Before we get started, make sure to like and share this video, smash the subscribe button and click the notification bell. Before discussing mini-brains, let us discuss the brain first, when it was invented and how it works. Fossilization of the brain, or other soft tissue, is possible, however, and scientists can infer that the first brain structure appeared at least 521 million years ago, with fossil brain tissue present in sites of exceptional preservation. The brain is an amazing 3-pound organ that controls all functions of the body, interprets information from the outside world, and embodies the essence of the mind and soul. Intelligence, creativity, emotion, and memory are a few of the many things governed by the brain. Protected within the skull, the brain is composed of the cerebrum, cerebellum, and brainstem. The brain receives information through our 5 senses: sight, smell, touch, taste, and hearing, often many at one time. It assembles the messages in a way that has meaning for us and can store that information in our memory. The brain controls our thoughts, memory and speech, movement of the arms and legs, and the function of many organs within our body. The central nervous system is composed of the brain and spinal cord. The peripheral nervous system is composed of spinal nerves that branch from the spinal cord and cranial nerves that branch from the brain. Now we have an idea about the brain, let us discuss. What are mini-brains and how to grow them in a lab with recognizable brainwaves? It may sound like something out of science fiction, but scientists grew mini-brains in a lab with functional neural networks that can produce brainwaves. These pea-sized lumps of cells don't look like much, but they are mini-brains, also called brain organoids, that were grown in a lab using human stem cells. And for the first time, scientists have created mini-brains with human-like neural networking capable of producing brain waves similar to those observed in premature babies. The work comes from a team of scientists at the University of California in San Diego who had their research published in Cell Stem Cell on Thursday. "We never had a brain organoid that can function like the human brain," said biologist and researcher Alison Mewattree. "The electrical activity of these brain organoids are emitting something we see during normal human development. So, it's a strong indication that what we have should work and function like the human brain." These brain organoids are still far from real brains, but according to Mewattree, it's a step toward growing a fully formed and functional human brain in the lab. Brain organoids could one day provide scientists with a clearer picture of what's happening during brain development. Neurological and psychiatric disorders such as autism, epilepsy, bipolar disorder, depression, autism, and schizophrenia are thought to have their roots in how our brains develop. It's not ethical to study during a pregnancy, so we have to rely on other models, mostly animal models, and those are far from the human brain, Mewattree said. And there are many neurological conditions that we know happen at these very specific stages. Most of the genes that are implicated in autism have a peak of expression, or activity, in the fetal stages, but you only see the consequences of behavior a little later in life. Let us discuss how mini-brains are grown in the lab. To grow a mini-brain in the lab, scientists take skin stem cells and reprogram them into pluripotent stem cells, which can develop into any type of bodily cell or tissue. From there, researchers place them in a cell culture that mimics the environment that allows our own brains to grow. By giving them a nice environment, the neurons will just thrive and make more synapses and take their time to mature and form these microcircuitries that the brain requires to have computational power, he said. How mini-brain shows the activity? Over the course of 10 months, the mini-brain showed consistent increases in brainwave activity or electrical signals triggered by firing neurons, similar to what's observed in the developing brain. At 2 months old, the mini-brains first began to emit some sparse electrical activity at a single frequency, like that seen in immature human brains. But by 10 months old, the mini-brains produced regular brainwave activity across a range of frequencies, similar to the brains of older fetuses or infants. It suggested that the mini-brains' neurons had matured and become interconnected, forming networks. Brain freeze, but, eventually, electrical activity in the organoids plateaued, as if they were stuck in infancy. The reason might be that you need more cells, or the conditions are not ideal, Muotri said. Or, it might be that the human brain, after 9 months, requires input. And the input further develops these networks. While more work is needed to understand how to take organoids to later stages of brain development, the team may have uncovered something that challenges previous beliefs about human brain development. The researchers thought fetuses in the womb picked up on sounds and other sensory information from the outside world that helped their neural networks to mature. But that wasn't the case at all for the organoids. What we've shown is that, no, you can have a brain outside the uterus, that has no stimulus from the outside, and it generates this sophisticated network activity. The brain is pre-programmed to wire that way, he said. Although brain organoids may behave like human brains in some ways, they're still far from the real thing. For instance, they lack nutrient-delivering vascular systems, and not all cell types are represented. And, as far as the scientists know, the organoids aren't capable of consciousness. But, creating a mini-brain that's conscious maybe isn't as far-fetched as it sounds. Mutori said the idea of producing an organoid with a neural network seemed impossible just 5 years ago. But given what he and his team accomplished, he's optimistic that scientists will grow brains that are increasingly human-like. I think we will learn how to make different brain regions and stick them together, a human cortex connected with human thalamus, together with a hippocampus. By sticking these pieces together, we may be able to reconstruct circuitries that are important in learning, for example. And we can then understand how the human brain learns, processes information, and stores memories. I think in the next 5 to 10 years, there will be a lot of advances on the frontier. Thanks for watching.